Hi everybody, it's Chris Torrance here. Just to give you a little bit of background on myself, my main experience is in cancer genetics and drug discovery, and I'm going to be talking today quite a bit about the full breadth of what we do at Horizon, not just about the gene editing, but how it can be used as well. And this kind of goes back to work I was doing as a postdoc at Hopkins with Bert Vogelstein. The technique you may not know quite as well as some other gene editing platforms out there. We've only been going about 5 years, so we're still relatively young. But that's changing fast. We're growing pretty rapidly, and the message is getting out there. And of course, the webinar today is designed to do that. Now, Horizon, I started for a very specific reason, and I think we're living in very interesting times where genomics, the rapid advances in genomics, can probably have a big impact on health and wellness and how we do medicine. But we are very much moving into the next phase. I mean, we have a lot of information, but not a lot of knowledge. And to turn that information into knowledge is going to require very good tools to understand what the genome does, what genes do, how they go wrong, and how we can target those things that go wrong to find new and better treatments. And, as I said, my background's in cancer genetics, and I think cancer is a great example of where genomics could play a great role. We understand quite a few genes go wrong now, but which ones are good, which ones are bad. And we also understand that we'll probably need lots of new targeted therapies, and we'll need to diagnose each patient for their particular array of genes that go wrong and tailor not just a single treatment, but probably a combination of treatments will be unique from one person to another. So this is a concept of personalized medicine. Now that's all great, but the speed that drugs are discovered is still relatively slow, and so that's really the driver while we started Horizon, is to see if we can try and speed that up, get new targets identified, get good drugs to those, and get them to the right patients at the right time. As we know, cancer is a moving target. It doesn't stay the same from one year to the next, and under therapy it can change very rapidly. So that's the sort of motivations to starting Horizon. And on the next slide here, I think we can sort of nicely summarize where the field is. We clearly understand that genetics has a major role in disease onset and progression, especially in cancer. It's very much a genetic disease. And we have a lot of technologies now that can help us understand what genes go wrong and even diagnose those in patients directly. The speed of next-gen sequencing, for example, is becoming really fast, and the cost is going down all the time. So that's really not a bottleneck anymore. I think where the bottleneck really lies is generating in vitro models that really sort of recapitulate very accurately the genetics of particular patient populations we're trying to treat. And I would hope by having better in vitro models that are predictive, we can decipher what genes do, how they go wrong, and actually test drugs in the laboratory against these patient models to see how they respond, which ones are the most appropriate to target in clinical trials. And we already know that there's enough proof of concept example drugs in there that are highly targeted to the root causes of cancer, that if you get them into those right patients that have that defect, you can see some pretty good effects. But we want to speed that whole process up. And I think one of the key bottlenecks is having good in vitro models that are predictive of what patients would do. And in order to generate these, what we're talking about there is a range of human cell lines that grow in culture that have particular genetic features that you can either characterize, but you will be limited on the number of lines that you can isolate directly for a diseased patient, and they'll always be quite complex in their genetics. So to have cell lines where you know exactly what gene is in there and what's gone wrong, and have a comparative normal cell line, this is going to require genome editing. And if you want to study pathways and things like this, you want to do things that don't actually occur in nature even. You want to tag a certain gene, you want to have a reporter to a certain gene. These things don't occur in nature. So very much moving into the realm of functional genomics, genome editing, to start deciphering the human genome. And coming on to Horizon and what we do, we're not just a gene editing company. We are based on a new technique that allows us to very reproducibly and precisely knock in or knock out genes in the human genome, endogenous genes. But then we use that technology in a very targeted number of ways to try and accelerate drug discovery. That's firstly building disease models that accurately reflect specific patient groups. But also we built up a lot of knowledge on how to study these excuse me, disease models, how to study those in vitro conditions that mimic the real situation in the patient's tumor. And we've built up a lot of knowledge that says that, by studying endogenous genes, they're not overexpressed. The phenotypes that one sees are often more subtle than you would expect. And you have to start to apply additional pressures, whether it be hypoxia or three-dimensional growth or some kind of nutrient starvation that tumors actually feel inside the body, that brings out why those genes were mutated in the first place. They're the selective pressures, and if you don't mimic those selective pressures in vitro, you don't see such good effects of targeted drugs. And this is a lot of know-how that's been built up based on these endogenous disease models. And we use these in a variety of different services, either profiling people's compounds or people buy the cell lines. We even custom generate cell lines of people. On the end of this continuum, you can see DX reagents. This is working very close to the clinic now. As I said, we'll need to diagnose in patients what genes go wrong in their tumors. And this is a very fast-growing field. We're not developing kits ourselves or assays ourselves, but we are providing genomic reference standards with particular mutations in the cell lines and putting those in specific ratios to start standardizing how well these tests work. So you can see we are much more than a gene editing company, very much targeted to applications as well as the gene editing. And the models we're creating and the custom services and all the ways these models can be used is reaching pretty good penetration now into both pharma and academia. We have a lot of clients, we have a lot of collaborators, and I'll be highlighting our collaborations as I go through this talk. So coming now to what the gene editing platform is, I'm going to, as I said, going to be giving overviews of the technique and overviews of how it can be used, and not getting into a lot of detail. There will be other webinars after mine over a period of time getting into more details and technicalities of everything. This is just a snapshot of what RAV is. You'll recognize that it has similar features to a technique that's been used for a long time that exploits homologous recombination, where you put in a DNA vector. It's a very simple vector. It's just a piece of DNA that has homology to your target locus. Now, when you use this approach in transgenic mice, you use plasmids, and that's fine in transgenic mice or even low organisms where recombination rates are quite high. But in human somatic cells, cells that are grown in the laboratory of human origin or actually mammalian origin in general, their recombination frequencies are very, very low. So those plasmid-based systems don't work very well. But if you put that same concept, that DNA vector, into a recombinant AAV virus, there's something about this virus that now reactivates homologous recombination in human and other mammalian cells. You get very efficient delivery for one, but we're starting to understand very well now that there's something unique in the fact that it's a single-stranded DNA virus, that this seems to reactivate homologous recombination in human cells and works about 1,000-fold to 10,000-fold better than plasmids. And this brings homologous recombination really into every person's ability in a realistic time frame, doing any gene anywhere in the genome. And you can also put in any type of change you want using homologous recombination. And the other great advantage of recombination is it's a very high fidelity DNA repair mechanism that we're piggybacking on, and so any changes you make are very precise. You put in the changes you want and nothing else. And we're not creating DNA breaks. I'll get into some of the other methods one can use, but we're not creating DNA breaks to increase recombination here, so we don't have the concerns that one might have by creating DNA breaks at your locus and other loci in the genome. As a technique, as I said, it's been around for a while in academia. We've not been around that long, so to speak. So this is really the first few years this has been commercialized. So you may not have heard as much about this versus some of the other techniques. And on the next slide here, I'm just sort of giving you a snapshot comparison to the other techniques that you may have heard about. Now, some of these are not going to be relevant to endogenous gene targeting. They're just there to provide a comparison. So things like overexpression and RNAi, they're very powerful, very high throughput, but they don't model the endogenous disease. So if a gene is lost in cancer or if it's mutated in cancer, these will only ever approximate it. They're overexpression models, or they don't model point mutations if it's an RNAi. So when it comes to modeling endogenous genes, we're really talking about two groups of technologies. One, AAV, which I just described to you just now, and the other, which are nucleases, and there's a few different nucleases. Zinc fingers have been around for the longest time. Megnucleases have been around for a long time, but they're not terribly flexible, not very easy to design a meganuclease to your gene of interest. Tau nucleases have been coming along recently that are very easy to engineer to your gene of interest, and as are zinc fingers, are relatively easy. Now their power, everything has pros and cons and nothing's perfect. From our perspective, our technique, while it's very highly precise, it's slightly slower. If you want to create knockouts, if you've got 2 copies of a gene, you need to do both those copies, you need to knock them out. And with our technique, it has to be sequential. So that's our detraction, is it's a bit slower compared to other techniques. If you're doing a single allele event, it's as fast as other techniques, and you have the precision. The zinc finger nucleases and the tau nucleases are very good in that they're very fast. They can cut 2 copies of the gene at the same time. Actually, if you've got even more copies than 2, which can happen in cancer, you can generally knock out more than 2 copies quite effectively. But that comes at the cost of precision. You are creating DNA breaks, and breaks will not be just specific to your gene of interest, but how often will that be a concern? And possibly not very often. Where they struggle a little bit compared to their abilities to knock out genes is to knock in genes. So you can provide donor vectors in trans with the nuclease, and at a certain frequency you'll get knock-ins. But with our technique, knock-ins and knockouts are equally efficient in both modes. Now, as I said, our detraction right now is it's slightly slower, but as I said also, we're still relatively young, and it's only early days of commercialization compared to the nucleases, which have been around for a while now. And I would say, they're probably reaching the zenith of what they can do. What they can do is what they can do now. Will they get tenfold better? I think we've got a lot of scope now. They're just AAVs just being commercialized on a grand scheme. We've spent a lot of effort and a lot of investment into finding new and better versions. We're already on version 2, which is substantially better than version 1. And we've got version 3 working now in our R&D department. That's giving us another roughly 20-fold increase in targeting frequency. So watch this space. We'll hopefully be launching new vectors soon that are even better than last ones. And I hope with this increased frequency of targeting that we'll start to be able to do sequential knockouts to allele steps very quickly in sequence, but perhaps even biallelic targeting to rival nucleases in their speed, but also have the precision involved with AAV. Now, moving on to what you can do with AAV, as I said, you can do the full gamut of things that you can imagine, both knocking out genes but also knocking in things very subtle. Our resolution is to a single nucleotide, and anywhere in the genome you can create any change you want. Now, we've been using it to model primarily patient genetics in cancer, and primarily those things are going to be therapeutic targets. So that's going to be generally point mutations that cause a hyperactivation of a target gene like a kinase. But we are using it in a functional genomics mode too. We simply use a selection marker to disrupt the coding sequence of a gene. More recently, we're getting a lot of clients asking to rescue RNAis, where you knock in silent mutations, multiple silent mutations at a recognition site of an SI or an SA binding sequence. And this is something that is very easy for us to do that other techniques find hard to do, to put in multiple subtle point mutations exactly where you want them. We, as I said, can do lots of other things. We can do large-scale gene deletions in one step. If you need really long-range deletions, you can do 2 steps by knocking in LOXP elements that are separated by megabases and then delete everything in between with a Cree recombinase. If you alter the orientation, you can do translocations. Amplifications we're also doing by knocking in ectopic DNA replication origins. And I would also say that when this technique was first used in academia, it was done primarily in the Vogestein lab, which is a colon cancer lab. And so the perception was that this was a technique that only worked in HD116 cells, and that's now been proved to be completely not true. Now we're commercializing it. We've tried a very large number of different parental lines, over 40 now, and over 500 different projects covering 16 different tissue types. So this works not just on adherent cells, works on non-adherent cells, works on ES cells and iPS cells. So it is a very broadly applicable technology to do disease modeling and functional genomics. Now, as I said, we're working very hard on next generation technologies, and I'm afraid I can't go into details yet because there's still research ongoing and still patents being filed on this. But it does involve broadly looking at more efficient selection strategies and signals that can potentially boost homologous recombination even further. The targeted frequencies now are on the scale of 25% of selected cells, which is about 20-fold better than previous. And I think this is really getting us now into the range where, well, certainly becomes very cost efficient to do, so prices will come down. We can do rapid double allele events and potentially one-step biallelic events now with that kind of frequency, and perhaps we can get even higher than that. We've got quite a big program of ongoing work in this area. And we can do scarless, depending on the approach you take. You can do LOXP, pre-excision, leaves a small footprint, but there are other ways of getting completely scarless with standard technologies. But if with having targeted frequencies over sort of 10%, you can think about dropping positive selection entirely and going completely scarless in one step. And this is something we're doing right now as well. Now, with those sort of frequencies, we could put a kit out there now with current vectors, and one differentiator of us from others is we actually do the work for you. If you've got a gene of interest, we'll generate the cell line for you. Now, we do have a sort of interim where, for academics who can't necessarily afford custom generation prices, we actually go out and teach them for free how to do it. But in the long term, to get wide market penetration, we realize we probably need to start putting kits out there. We could put out kits now, but I really want to put a kit out there that is foolproof, very high efficiency, and thus very cheap for you to get your gene of interest knocked out. And I think we'll be very close to that with some of the new advances we're working on now. Now, moving on to the applications, and the first application really is generating these disease models. And we have our second core product, so to speak, are the X-Man cell line range, and this stands for gene X, mutant and normal. So these are isogenic pairs of cell lines that are genetically identical but only differ at a particular gene locus of interest. And so you have a very definitive pair of cell lines that tell you exactly what your genetic alteration does compared to a reference normal cell. And we can actually start from normal cells and build tumors from the ground up. Or we can go the other way, find a cancer cell with a gene of interest, a mutation of interest, and correct it back to normal. So you can study both ways. Either way, you've got isogenic pairs that give you a very definitive system to study gene function and even drug effects on that particular cell with that particular mutation. Now, we've got a very large bank of cell lines. Because of our background in oncology, we focus mostly on oncology to this point, but we are very interested in moving into other therapeutic areas. And this is where our centers of excellence comes in, which I mentioned previously, is trying to get this technology out far and wide firstly, but also trying to get other therapeutic areas covered as fast as possible. We're experts in oncology, not so experts outside of oncology. In the centers of excellence, what we're doing is we're trying to identify labs that have been core facilities, for example, doing mouse transgenics and getting them to generate human disease models. We're also working with individual labs as well, trying to get them working in gene targeting if they've never even done gene targeting before. And we're starting to get quite a few lines coming through now in a variety of different areas, from neurodegeneration to diabetes, all sorts of different therapeutic areas. And we're also very involved in collaborations. We're part of several grants that allow us to stay at the cutting edge of understanding what new genes and biomarkers are coming out in particular disease areas to actually go and generate disease models about. There's ColThreads, which is a colon cancer consortium, Predictor, which is a kidney cancer consortium. 4D Cell Fate is a consortium trying to understand embryonic stem cells and the differentiation patterns, understanding how the histone methylases and demethylases work. So moving on now, the last area, this is a very new area we're looking at, is bioproduction. This is moving on the concept of, human cells and mammalian cells are used to create biotherapeutics themselves. A CHO genome has just been done. This is a very common mammalian cell line that's used to create biotherapeutics. Now, the yields that one might get of a gene of interest could be low, or perhaps it's hard to purify. Perhaps your gene gets degraded. You may need very specific glycosylation forms. These things are hard to do unless you engineer the parental line in some way. You can knock out genes that restrict purification or cause too much glycosylation in a certain direction. You can insert your gene at specific landing pads, so you know you're going to get consistently high expression from that locus. You can even retrofit cell lines that were created through random integration, but you've just naturally selected a particular parental line. You're very confident in its properties, but now you want to put your gene of interest into that same locus, so you can retrofit those by producing lines too. We've created a first proof of concept with a glutamine synthetase knockout, which is a selection gene that's used to put in your gene of interest. By removing it from the endogenous genome, it allows a greater selection pressure. This is something the field's needed for a long time, but there's plenty of other things this field needs, and we're working very closely now with a CRO and a major pharma in engineering CHO lines in other ways. We're going to be looking at how disease models can be used to speed up drug discovery at several stages. As I said in the intro, we're all about trying to model patient genetics in a test tube in a very accurate way, and then use these better in vitro models that are predictive of patient genetics and how they might respond to new targeted therapies throughout the drug discovery continuum, from the earliest stages of target identification and validation to actually profiling early drug candidates for are they showing a selectivity window for tumor versus novel as we'd hope, to the later stages of I've got a drug candidate, which are the most relevant patient populations to study. Now we've got a bank of over 500 different disease models, and we have literally had people say, I have no idea which patient group to go after. I want to do a selected clinical trial. Can I just throw them everything in your bank? And we've done that and successfully identified patient groups that they've gone on to design trials around. And I'll take you through examples of all these data as I go through. Now, before I get into specific examples, you're probably gaining an appreciation of a number of ways we can work with people. We can generate custom cell lines. We provide cell lines off the shelf. We also have a route where you may have a very specific question. You may have broad-ranging questions, and therefore you license the lines or a large bank of lines. But if you have a very specific question and you want to address it across a wide range of patient genotypes, a very convenient way of doing this is to send us few compounds and we'll profile them. So we have a CRO division that is expert and fully enabled with the full bank of 500 disease models that we have, and they can be wide profiling experiments, or they can be more bespoke experiments where you study particular targets, and we can do that with compounds or siRNAs or shRNAs, or doing downstream analysis, looking at pathway analysis, Western blots, mode of action studies with apoptosis, autophagy assays, a variety of different assays. Or we can build high-throughput screening tools, putting reporter genes downstream of a gene of interest so that you can do your own high-throughput screens. Complex high-content imaging platforms, they can also be relatively high-throughput, all the way through to in vivo modeling. We work with a partner here called Crown Bio. They're fully enabled with the bank of cell lines that we have to grow xenografts. So we can offer the full gamut of profiling capabilities in a CRO service, as well as you licensing the cell lines. And I'd say another way that we can work with clients is this is very much based around the concept of, as a field in general, especially in cancer, we're very much moving into a new brave world of we've got lots of target candidates, but which of those are really truly good targets? The low-hanging fruit, so to speak, is being worked on for cancer, and now we all need to find new targets. And that's exciting, but it's also quite risky. And so spreading the risk a bit is we're working in a very collaborative way now with a number of different companies where you use our ability, our unique ability, and our ability to understand gene editing in general to take your target of interest and precisely understand what it does and is it relevant to your disease area. So that could be knocking out your gene. Does that have a lethal effect on cancer cells of a particular type? Or putting in subtle point mutations. That could be, let's say it's a kinase, could be a kinase dead mutation to mimic what a compound does. These things you can do very accurately and precisely with genome editing. But then we have the full gamut of services to take those disease models and support drug discovery itself. If you're a relatively small company, we can do those, support you on the biology side as well as chemistry. If you're a virtual company, all the way through to in vivo modeling, as I said, with Crown Bio. Putting that all together as a package, people access it on an FTE basis because it's a piece of work that can change over time and roll over a period of time. And we would take a position of risk in this as well, where you don't pay such a high price for these custom generation services or CRO services. You pay a reduced fee where we would take some upside in whatever we find. So we validate a target and it goes through into drug discovery and it becomes a drug. We would take milestones through that process. And that's just a different way of working. It's a lower upfront cost but brings some risk share where we would share in the upside if it was successful. And we are working for several companies in this fashion now. Now coming to the specific examples of what we might do in target identification or actually drug discovery throughout the rest of the drug discovery continuum. Starting with target ID and validation, here's an example in a collaboration with Celera where we take a pair of isogenic cell lines that differ only in mutant PI3 kinase. So this is a very commonly mutated pathway. People are trying to target it directly with PI3 kinase inhibitors, and I think that's a good idea. But are there other things that mutant PI3 kinase does that could be good targets? And metabolism is a very new area that people are quite excited about that could bring very good tumor-specific target aspects over normal cells. So we can take a normal cell that's got no mutant PI3 kinase and an isogenic variant that's got mutant PI3 kinase and just expression profile. And we've got very interesting differences at the gene expression level in the metabolism genes. And PI3 kinase is one that's very cleanly upregulated in glycolysis. Other genes we study, genotypes we study like KRAS and p53, they upregulated different aspects of metabolism. And when we went to study those effects, are they creating gene addiction scenarios, they didn't hold up. But in PI3 kinase, the gene addiction scenario that's been suggested here, glycolysis would be highly dependent, really is true, and that dependency is very robust. So if you start to grow these cells in anything other than glucose, you see the data on the bottom right here, the cells really don't like it. And if we move now to further validate this hypothesis that okay, perhaps I can target something in the glycolytic pathway such as hexakinase 2, which is one of the most highly upregulated genes in a mutant PI3 kinase cell line. When you knock down hexakinase 2, you see similar effects to starving the cells with glucose. Very high dependency on that pathway. And that's gene expression analysis, providing an example. What you could do is use SI screens directly, so instead of just validating results, you can actually use them in functional genomic screens. And this is something we're doing in our synthetics program. Synthetics is a concept that we're using to try and deal from the cancer genome. For those who are aware in the cancer field, I sort of alluded to it earlier, that we're information rich but knowledge poor. So we've got a lot of candidate targets, but which of those are really real, and which of those that are real can we actually do something about? And the answer to that is frustratingly little. So most of the genes that go wrong in cancer are either lost entirely, so they'd be tumor suppressors, but many oncogenes even, things like beta-catenin and KRAS, they've been perennially hard to drug, so they're not classical kinases that we can drug very easily. So we're going to have to take a very systematic approach next to try and understand what those genes do, what they regulate. That if we can understand what they regulate, we can maybe find druggable nodes downstream. But also in the process, in doing functional genomic screens, perhaps we can find things that are completely unpredictable. And this is the idea of sort of synthetic lethality, that with the right disease models, we can screen biology directly for unique and sensitive points that tumors are sensitive to that normal cells are not. And these nodes of sensitivity may not be completely predictable, but we don't have to predict them if we do cell-based screens, and we do them in a systematic and high-throughput way. So this concept of synthetic lethal screening has been around for a while. I'd say it's probably been done not quite as well as it could be, in the sense that if you use non-isogenic cell lines, which most of the field's been using, you use large panels of unrelated cell lines, and you try and correlate the differences in cell effects you might see against an SI library. Now, it may be to the gene of interest that you are trying to target, but it could be to any other genes that are in there too, because there'll be thousands of differences from one cell line to another. And perhaps even it's very hard to represent your gene of interest in a panel, even if it's very large. As we're moving into less common genes in cancer, it is actually very, very hard to find any cell lines represent your gene of interest. So moving on to what we can do, and our unique selling point in this respect is we can model any gene of interest, any variant with any gene of interest, and provide a very simple binary system that has either a wild-type version of your gene or a mutant version of your gene, and screen against the SI or SH libraries of interest, the things that only kill the mutant, not the wild type. And that could be gene A, B, C, D, whatever. Now, I think previously one of the reasons why non-isogenic panels create a lot of noise is you're combining two very noisy systems, a lot of different unrelated cell lines with quite a noisy system in RNAi itself. They're good, they're high throughput, but they have off targets. So if you combine off targets of the SIs with the vagaries of unrelated cell lines, I think that just becomes a little bit too much. If you combine isogenics now, I think there's a lot greater potential to find hits that are really real. And there's a good example of synthetic lethality, probably everybody knows now, is the BRCA-PARP interaction. This is data from Alan Ashworth's lab. It's quite old now, but I put it up there as a good example of what one can do with synthetic lethality, that this concept that while cancer cells do things to allow themselves to mutate at high rates, such as losing DNA repair genes, this does open up vulnerabilities that just aren't there in normal cells that one can exploit. And if you look at the bottom right-hand panel, the kind of differences you see with a PARP agent in cells that are wild-type versus BRCA2 null, we're looking at black and white situations here. Now, pathway-targeted agents going after gene addiction can approach this, but often it's very hard to approach that kind of tumor versus normal selectivity with pathway-targeted agents. Plus, the compensation pathways that occur are very rapid. So I think there's real complementarity needed to take this approach of synthetic lethality. But the big question is, how much more will we find? But that's what we're going to do, and we're taking a consortium approach to do this, with the ICR, Alan Ashworth himself, who's involved in this program. But from a practical perspective, the work is being done at Horizon. We're doing an SI approach. And the other partner that's in the consortium right now is H3 Biomedicine, and they're going to take an SH screening platform. And the idea is to combine both these platforms to screen 50 undruggable cancer genotypes that represent, in many cases, very common genes that are undruggable, things like P53, P10, but also genes that people are trying to target but perhaps not had a lot of success, things like KRAS, and throwing at them candidate targets that are druggable, kinases, epigenome, metabolome, ubiquitinome, things like this, and just finding things empirically that selectively kill the mutant cells versus the wild-type cells. This is going to be a 3-year program. We anticipate we'll uncover quite a few targets, possibly up to 100 new targets. People in the consortium will get preferential access to these targets and early access to these targets. We imagine there'll be quite a surplus. These will be available for people to license outside the consortium, but we are looking for other consortium members to join at this stage. We've got another big pharma very close to joining. And when you join, as I said, you get preferential rates over those targets, but also early looks at what these targets are looking like as they go through the validation cascade. And a target that's come out of this program already that sort of demonstrates very nicely what we can do with synthetic lethality over and above the history is a new target. I can't reveal what the identity is right now. This is called HD001. It's a target that does target KRAS activation. It seems to have synthetic lethality with that genotype. The target itself is also mutated directly in cancers, and these cancers also seem to be hypersensitive to knocking down or knocking out this target. Now, the target, I can say, is a kinase, so it's very druggable. And we are actually drugging it. We've had a chemistry program going for the last 10 months. What we're not trying to do in synthetics is become a pharma company. That's not our role. I see our role being feeding pharma with new and validated targets. Now, part of that validation, I think, is to do some early chemistry against these targets to show you can do the same thing with a small molecule that you can do with a genetic tool. Genetic tools can be very clean, as I showed you with the RAV technology, but small molecules are not. And so can you recapitulate those results? So we need to take it down that road a little ways, but we want to partner at the LO stage on most of these things coming out of synthetics. And this is where we are now with HD001. We've got several series with very low nanomolar potency. They are also having good KRAS synthetic lethality, and we're very close to partnering this one now with a major pharma. And we just aim to do this over and over again against all the other undruggable genotypes in cancer. So moving now into other ways that we're actually helping companies right now. If we can screen lots of SIs and SHs, perhaps we can screen lots of compounds as well, and we certainly can, and we are. And this is ideal now to start tracking biological SAR. So not just doing SAR against your target in a test tube, like biochemical target in a test tube, but understanding what your compounds do. They may be very potent against your target, but they will hit other things as well. Will they give the selectivity windows that we anticipate in biological systems? Now, it's very easy with isogenic systems to get definitive readouts of a tumor normal selectivity to a particular genotype. This is what we're doing here. This is proof of concept data we did for a client, looking at some early PI3 kinase inhibitors, comparing them to a gold standard PI3 kinase inhibitor from Genentech that's now in clinical trials. And you can see with the gold standard inhibitor, you can see a nice window of selectivity for mutant cells versus wild-type cells. And here are several other series from a client where there are similar levels of potency against the target but different selectivity profiles, trying to understand which series is most relevant for them to continue. Clearly it's going to be the one at the top left that's starting to show windows of selectivity. Not quite as good as a clinical candidate, but it is giving you an early look at biological SAR and helping you hone in on the right series to study. And I'd say this is exactly what we've done too in our HD001 program. Now, those windows of selectivity are reasonable, they're nice, but I mentioned right at the beginning our expertise in studying the tumor microenvironment in vitro and how this is really required to see the true effects of targeted agents. So if we stick with PI3 kinase as an example, on the previous slide we can see selectivity windows. They're nice and they're reasonable, but are they as big as we really should see for a targeted therapy? Perhaps, we're studying the wrong phenotype, and this is kind of what we're studying here. We put the same cells into now a 3D matrix. So previously they're growing in 2D, flat on plastic with lots of nutrients, etc. If you put them into a 3D matrix where they've now got to survive in a sort of harsher environment, they've got to grow and invade through a solid matrix, you can see a very different phenotype now of mutant versus wild-type cells. Previously we saw no difference in growth rate in 2D. Now we don't necessarily see a difference in growth rate in 3D, but what we do see is a stark, dramatic difference in phenotype in invasion. So the parental size grows tight balls of cells, whereas the mutant cells grow these highly branched and networked and invasive like cells. And if you come along now with a targeted inhibitor, you completely reverse that phenotype, which is a black and white phenotype, and you've completely reversed it. So much bigger windows of effect here. And this was work done in collaboration with Genentech, and they've really characterized very nicely the gene expression level and the morphologic level and the EMT level, showing that this really is a metastatic phenotype that PI3 kinase is primarily driving. As I said, we're not driving growth, primarily just metastasis. And when you study compounds under metastasis, you see a much bigger effect than you do on growth. Now, moving to sort of later stages of drug discovery, as I said, we've had people come to us and just say, I've got a candidate, and throw against everything in the library. And this is some example data where looking at a range of gain-of-function point mutations in a common set of parental cell lines, we can see the effects of a targeted inhibitor to EGFR. In this case, it's the EGFR mutant cells that are by far more sensitive than any other cells, including the parental cells with no mutations, but certainly the other cells with other mutations. And actually, you can take the EGFR mutant cells and knock in an additional mutation, such as PI3 kinase or BRAF or KRAS, and they'll turn very sensitive cells now into completely resistant cells. And this is exactly what we're seeing in the clinic as well. So we can model both retrospectively here known sensitivities, biomarkers that dictate sensitivity, but known markers that dictate resistance. And I'll show more evidence of this as I go through. Now, one of the great attractions of doing gene editing is you can do a lot of different things very, very quickly. You may be able to find cell lines that are EGFR mutant, you may be able to find a couple of different variations, but if you want to look at all the genes of interest and all the possible variations, and for EGFR there's literally hundreds of different variations, it's impossible to find cell lines that cover all these things. And they will be non-isogenic, so they won't be directly comparable. To get isogenic cell lines with all the different variants, it's very easy for us to engineer those. And you can start to see subtle but real differences in the isogenic settings between different variants. So if you take EGFR as an example, the blue parental lines here, the deletion mutants seem to be the most sensitive to EGFR target inhibitors, very closely followed by the A5R mutations. Somewhere in between is the 861Q. T790M, we understand, is a resistance mutation, and clearly here we're seeing nice resistance compared to the parental lines and the other sensitive lines. If we take BRAF as an example, so while the EGFR data we know is true, it's retrospectively, we can compare it to clinical data, here's a more prospective look where we don't quite yet understand from the clinic what might be happening. Now, there's a very common variant, V600E, and in the clinic, melanoma patients with V600E seem to be doing very, very well, at least for a period of time until resistance kicks in. And we've modeled that quite nicely. Now, there's a less common variant, V600K, and we don't have information yet from the clinic how well they respond, but the in vitro models are maybe suggesting that they might not respond quite as well as a V600K. So this is certainly something to look at, to suggest something to look at in the clinical setting. Here's another example with IDH1 mutations and IDH2 mutations. IDH is a very big gene now, lots of people working on it, trying to find targeted agents. There's the concept that all mutations induce a biomarker called 2-hydroxyglutarate, and using an isogenic setting, we've demonstrated that not all variants are in fact producing 2-HG. The R140Q mutation in IDH2 is not producing as much 2-HG, anywhere near as much as the others. And this only became apparent when we studied expression at endogenous level. People have studied overexpression systems, not seen a great difference between different variants, but that's because you're saturating the system, showing the importance, really showing the importance of studying things at the endogenous level. Now, moving on to sort of resistance biomarkers, I've alluded to how this is a big issue in cancer now, that resistance mechanisms are very common, and this is showing how prospectively how the models are predicting patients too. So all the other data is kind of retrospective, but this is really prospective. Now, there's a long story, and I'll make it short, that KRAS is a well-known biomarker to resistance to EGFR therapies. At the moment, people think that all the variants of KRAS impart resistance. Now, the in vitro models suggested that the G13Ds probably were sensitive, and based on the in vitro models, clinicians now have gone actually prospectively into patients and demonstrated that's true. So really got very strong evidence now that these models are predictive of patient situations. And we've extended it now to other targeted therapies, not just EGFR therapies. KRAS seems to be imparting resistance to many different targeted agents. MEK inhibitors are shown here. And I'll just focus on the bottom panel here, showing how another great use of the models is to find rational combinations. So if target therapies are resistant, here we can show KRAS G12C is resistant, but if we give a lot of combination with cetuximab, we see those lines become sensitive as the original parental cells. And this is starting to be defined now, that things that target the KRAS pathway very quickly cause a de-repression of receptor tyrosine kinases. This could be the EGFR receptor or other receptor tyrosine kinases. So this is explaining why that particular combination works very well. And all this is being studied within the collaborations I told you about. And a last way, when you actually do have a drug candidate, is you want to get as much mileage out of it as possible. So this idea of drug reprofiling is becoming quite common now. And we can do this very quickly with a large array of patient-relevant models, as I said. Here on this slide you can see a couple of screens where we're trying to find things that target PI3 kinase and picking out things that you wouldn't have expected. Things like anti-inflammatory drugs have a selective activity for mutant PI3 kinase cells. And here on the right, how lithium chloride, a known GSK3B inhibitor, seems to have, again, specific effects on mutant PI3 kinase cells. And this one's actually being progressed now into clinical trials because it is already used for bipolar disorder. And the last wave is really right at the end again of how we can help the drug discovery continuum. This is actually in the clinic now. I alluded right at the beginning of how we're trying to standardize molecular diagnostics. We can do this in a couple of ways. These kits, there are a variety of different kits out there, and next-gen sequencing is also being used as a diagnostic tool, but we don't know really what the limits of sensitivity of these various platforms are. Plus the biopsies you get out of a patient is quite a mixture of tumor and normal, and sometimes it's actually a minority of tumor cells in there compared to normal. And when can you actually not detect your gene of interest? Tumors are also very heterogeneous on their genetics. So there's a real need to standardize in your test, have a reference mutation in the test alongside your test sample, but you know exactly what gene's in there and you know what ratio of mutant versus normal there is. Now we can create these stoichiometric ratios of either gDNA directly, but also put in ratios of cells into paraffin blocks, so that this goes through the full extraction procedure one would normally use on real patient materials to understand not just the sensitivities, but the processing, how efficient and how good that processing is. The last thing I'm going to talk about now, outside of disease models, is our ability to model pathways. And I think this is a new area. We've got several genes covered right now, and you can see them on this slide here. We want to model pathways for a couple of different reasons. Perhaps early stage molecules before their drug candidates hit a number of pathways, and perhaps having a sensor for all these pathways would be good to help you hone in on a specific one. But also you could have high throughput screens to particular targets that aren't classically druggable, or targets that have to be assayed in live cells because you can't recapitulate the full complex of things that would be, for example, on a transcription factor. Now, whatever approach you want to take, you've got to try and be very sensitive, because we are trying to model endogenous genes here. And to detect endogenous levels of proteins or promoter activities, we really thought we had to move beyond things like GFP, which are very good, but are they sensitive enough to register endogenous genes? Now, HADERtag and nanoluciferase are two new products from Promega, who we're working with very strongly with for a number of years now to try to marry endogenous gene editing with high sensitivity reporting technologies. HADErtag allows us to very sensitively understand protein localization in live cells, or to pull out a target and do proteomics to see what else is bound to it. Nanoluciferase is a way of developing homogeneous assays to register either promoter activity or protein levels to have it in a high throughput screening format. Here's an example data for HADErtag showing how you can do live cell imaging using a ligand to HADErtag that fluoresces. Here we've got a halo tag appended to KRAS at the endogenous level, and as you would predict, seeing that KRAS is nicely associated to the membrane. And using this as a pull-down technique, I showed you data earlier that suggested that different KRAS variants have different effects on therapeutic response, and maybe different variants of KRAS go through different pathways. We start to address this by actually pulling out these different KRAS variants now with halo tag ligands and running mass spec proteomics profiling. Here's an example with the wild type where you pull out the wild type and you can pull out all the proteins bound to it. You pull out things that you would expect in the KRAS BRAF pathway, but you pull out a lot of other things you wouldn't necessarily expect. Here are a lot of other genes involved in mitochondria. And when we analyze this same approach using the KRAS mutant variants, you see some similarities. You see some new things that the wild type doesn't bind. But one thing you see very clearly is that all these mitochondrial genes interactions go away. And this could be starting to address the Wahlberg hypothesis, how mutant oncogenes activate glycolysis. If you're no longer binding to mitochondrial proteins, somehow you're dialing up glycolysis and dialing down the TCA cycle. And this is work we're following up on. Coming to the nanoluciferase, nanoluciferase is a new version of luciferase that's much smaller, much brighter than the old luciferase. It's about excuse me, it's about 200-fold brighter than the old luciferase. And we can see here some proof of concept data where we've knocked it into endogenous promoter. Here on the left, HIF1 alpha, and on the right, P21. And we can see very nice signal to noises here, even at the endogenous level with nanoluciferase. And the signal to noise is good when we inhibit it with a general transcription inhibitor. You can see reduction of a high signal down to 0. In the case of P21, where it's actually a gene that's inducible with DNA damage, if we give a DNA damaging agent, you can see a nice induction of P21 prior to any overt cell death, which is registered by simple titer glow reagent. At the protein level, we can see very good signal to noise as well. This case, using NRF2 as an example, we don't have a specific inhibitor of NRF2 protein levels, but if we just use cyclohexamide, we can see over a period of time, because the half-life of the protein here you've got to take into account. At higher time points, you can see a nice reduction of the signal in NRF2. We can also stabilize that artificially with a proteasome inhibitor. If we go to the next slide, we can, looking at the HIF1 alpha gene, we can stimulate its levels in a more specific way using hypoxia. And this slide is also demonstrating one of our ambitions of putting these reporter lines in the high throughput screens, where we've taken the HIF1 alpha reporter cell line and screened that against 1,000 SIs to a variety of different targets, kinases, ubiquitinases, epigenome targets, and trying to find things that can cause a shutoff of HIF1 alpha without causing overt cell death, so we know it's specifically transcription that we're affecting. Now, of course, a positive control here would be HIF1 alpha. You can see that nicely shuts off HIF1 alpha protein before any effect on viability itself. A positive control that will kill cells is polokinase 1. You can see clearly it's killing cells, and it's not through a transcriptional effect here. The reduction in transcription is purely through killing of cells. And if we take the rest of the library, and I'm just showing you the interesting hits we get here, you pick out things that you would predict, things in signaling pathways that are known to regulate HIF1 alpha, where you see knockdown causes significant transcription shutoff with minimal effects on cell viability. I'd say the PI3 kinase gives a better effect in this respect of shutting down transcription without affecting cell viability. The KRAS pathway does this reasonably well, but here are some more novel targets that do this quite well. SIRT5 is probably one of the best. We get very good shutoff of the HIF1 alpha protein levels with very little effect on cell viability. So this is just proof of concept screening. 1,000 is still not that many genes, but we're working now with a company that's starting to run very big screens using SIs against this cell line now. And the last way we're using luciferase is in vivo imaging. Now, I'd say all the in vitro applications, the cell lines that I showed you, and the reagent to do the luciferase imaging and halo imaging, these are all products that are available off the shelf now. Something that this isn't available yet, but it hopefully will be soon, is the in vivo imaging, where we're just using luciferase here as a surrogate to register cell number without having to physically measure the tumors. Here we're just looking at xenografts growing on the skin of the mouse. But the real aim is to try and to register models that you can't access, orthotopic models, metastatic models, to look at tumor burden in mice without invasive techniques. But here at the xenograph level, you can see it working very nicely. This is measuring the tumors, and this is using luminescence from luciferase. You can see a nice concordance there. You actually see a drop-off of very large tumor sizes, and I think this probably has something to do with the tumors being necrotic on the inside. If you, on autopsy, you see they're actually dead in the middle as they're getting bigger. So that's one attraction, even in a xenograft setting, is that if you just measure them, you think the tumors are still growing, but if you use imaging, they're actually dying on the inside. So now that's coming to the end of the presentation. I try to give you a very broad brushstroke, as I said at the beginning, of what Horizon does. It is based around a core engineering genome editing platform that's very precise, allows you to knock in and knock out any gene in the human genome or other mammalian cell lines and do any alteration you want to a single nucleotide resolution. We've built up a very large bank of disease models already, around 500 different models in a variety of different parental cells. We do custom gene editing services. We also have a CRO division that can profile compounds and even do target identification and validation in our Synthetase platform. All the way to the clinical setting with Horizon Discovery Services and producing bioproduction biotherapeutics in bioproducer cell lines to high yields and higher conformity by knocking in and knocking out genes in CHO cell lines. And so that's the end of the presentation.